So here's the image that Erica was describing that is a composite of several different images. So I'm going to tell you not just about how I made this image, but also about how the information in this image is helping us make advancements in vaccine research. So if you look closely, you'll realize that this is actually made up of thousands of smaller images. These are images of lymph nodes, which are the organs where the immune system learns how to mount a response to disease. You might be familiar with the sore feeling of swollen lymph nodes that can happen during an illness. Lymph nodes swell when the cells from the immune system start multiplying in an effort to fight whatever pathogen is causing the disease. So for example, if the disease is being caused by a virus, some of the cells in lymph nodes create defensive molecules called antibodies. Antibodies can attach to the virus surface proteins to block the virus from functioning properly, and also to mark the virus clearly as something dangerous that needs to be destroyed and removed from the body. The way that vaccines work is to train the immune system to recognize a virus and to create the correct neutralizing antibodies without you having to actually be exposed to the real virus or to getting sick. The training material, or the protein that we want antibodies to recognize, is called an antigen. So for the immune system to properly develop antibodies against an antigen, it's important that enough of the antigen gets into lymph nodes so that the immune system can learn about it. For my own project testing new HIV vaccine designs, I modified an HIV antigen with a fluorescent dye to make it glow under certain kinds of light. This modification allowed us to track the antigen to see how much is getting into lymph nodes, and this is one of the methods we use in the Irvine lab to compare and contrast different vaccine designs. As shown in this Venn diagram, we can experiment with vaccine designs in a variety of different ways, including new types of antigens, new types of adjuvants, which are materials that help activate and turn up the immune system's response, and even new methods for delivering vaccines. So our lab has many different projects that are testing these aspects of vaccine design, as shown here, including some that combine more than one area. But to bring it back to my image and the reason that I was looking at so many lymph nodes, I want to tell you about one of my projects in particular, in which we compared 3 different methods for delivering vaccines and tracked how much antigen would get into lymph nodes with each method. So traditionally, when you get a vaccination, you get a single shot called a bolus injection. We wanted to compare this to what would happen if we were to split that same total dose into a series of injections over the course of 2 weeks, or even to use a device called an osmotic pump that would deliver the same amount of antigen slowly and continuously over that same time frame. However, when we got the samples back, the really frustrating thing is that we had all these lymph nodes that looked essentially the same from the outside. So I had no immediately obvious way to know which samples to spend my time looking at to see where the antigen was. So after spending a couple months doing trial and error with this, I eventually found out about a special kind of flatbed scanner that's not typically used for looking at tissue samples like this, but that I knew could detect fluorescent dyes, such as the one I put on my antigen. So fortunately, this worked great, and it could detect the fluorescence shining through from the inside of the tissue. I could then take these images of the lymph nodes from the scanner and color them with a heat map artificially. So the bluer end of the spectrum indicates low levels of antigen, and the redder end indicates more antigen. And this was a real life changer for me because it let us see right away that there were interesting differences between the samples from each of these different methods of vaccine administration. So as you can see here, there seemed to be much more antigen getting to the lymph nodes when we deliver the vaccine slowly over time compared to the single shot. So in order to create my composite image, I wrote some code to just sort all of the lymph nodes that I imaged like this from multiple different experiments, not just this one, and to put them in a giant grid with the brightest ones in the center. And like some of these other talks, that was just to make a pretty picture, but the source images in this case were actually very useful in themselves. So if you look even closer, you'll notice that the antigen that was delivered slowly over time has accumulated inside this lymph node in a really interesting way. So in fact, this pattern of antigen accumulation had never really been seen in this way before. So images like this from the flatbed scanner allowed us to focus on interesting samples, such as that one, and that therefore enabled me to use other kinds of fluorescent microscopy to confirm this unique antigen accumulation pattern and to see it in 3 dimensions that's shown here. The really interesting thing to me, though, is that we don't actually see the same pattern from the antigen delivered by the traditional bolus injection method. So that's shown on top here, compared to the slow delivery method on the bottom here. So this result is actually really exciting because in a parallel experiment where we tested the antibodies that were produced in response to these different forms of vaccinations, only the slow delivery methods produced antibodies that could actually protect against HIV. So these images are actually helping us figure out how that happened. And so they're helping us get one step closer to making a working HIV vaccine. And to bring it back to COVID, if that wasn't exciting enough, we think that these improvements to vaccine technology should be useful in designing vaccines against many other diseases, including the coronavirus. So with that, I'd like to acknowledge the support of my postdoc advisor, Daryl Irvin, the technician who helped me on this project, Ben Cassette, who's now a PhD student at Duke, and the rest of the Irvin Lab, the core facilities at the Koch, my collaborators from other institutions, and the funding sources. And I'm happy to take any questions.